If two of the most trusted brain scans for detecting Alzheimer's disagree more than half the time, which one should you believe? The answer to this question just changed everything we thought we knew about early detection and could mean years of additional intervention time for those of us at risk. Hi, I'm Dr. Kevin Tran. I'm a doctor of pharmacy and a double APOE4 carrier. I'm the founder of the Phoenix Community, a platform dedicated to helping APOE4 carriers beat the odds and outsmart Alzheimer's. So through my personal journey with APOE4, I dive deep into the latest research and translate complex findings into clear, actionable insights we can apply today to take control of our brain health. And today, I am bringing you revolutionary findings from the Alzheimer's Association International Conference that happened in July 2025. 6 research teams just presented discoveries that challenge decades of assumptions, from brain scans that contradict each other to the shocking revelation that our brain might actually grow larger before Alzheimer's takes hold. These aren't incremental findings, they are more game changer about how we detect, understand, and potentially prevent cognitive decline. Let me start with something that affects all of us, but that most people have never heard of: the brain's clearance system. So this is known as the glymphatic system, and Dr. James Lefebvre from Vanderbilt just revealed how we can now see when this crucial system starts failing, potentially years before memory problems begin.
Perivascular spaces are small fluid-filled channels that surround cerebral blood vessels and facilitate the clearance of waste from the brain. Enlarged perivascular spaces represent the enlargement of the perivascular space. Increasing evidence suggests that they indicate reduced waste clearance and contribute towards cognition.
Right, think about this for a moment. Our brains have their own drainage system, like tiny pipes running along blood vessels. And these work primarily when we sleep. So when these pipes get clogged or enlarged, so waste products that include amyloid beta and tau protein that should basically be flushed out of your system, while they start accumulating. So these enlarged spaces visible on standard MRI scans are most common in 2 critical brain regions: the basal ganglia, which controls the movement and the habits, and the white matter, which acts as the brain's information highways. And now, for the first time, Dr. Lefebvre's team has developed an automated tool called DORS that could detect these enlarged spaces with unprecedented accuracy.
Both basal ganglia and white matter, DORS exhibit a statistically significant higher dice score compared to other tools. Both basal ganglia and the white matter, DORS exhibit a statistically significant higher F1 score compared to other tools.
So let me decrypt this a little bit. The DICE score and the F1 score that Dr. Lefebvre mentions are measures of how accurately the tool identifies these spaces. Essentially, DORS catches what other tools miss while avoiding false alarms. This means we can now identify when the brain's cleaning system starts failing, potentially years before symptoms appear. So for us as APOE4 carriers, understanding how our brain's waste clearance could be crucial, since we already are at a higher risk for protein accumulation that defines Alzheimer's. So our genetic variant affects how efficiently our brains clear these toxic proteins, making these early detection of clearance problems even more critical. All right, next, let's turn to Dr. Andrea Rocca's presentation from the University of Pittsburgh, which exposed a fundamental problem in how we detect tau tangles, those basically twisted proteins that damage brain cells in Alzheimer's. Her team compared the 2 most widely used tau PET tracers, which are MK6240 and Flortaucipir, in the same individuals from the HEAD cohort study, which included around 500 participants. And what they found was shocking.
When we pair the individuals head to head, the concordance is only 47% between tracers. So only half of the cases, the Brack stages agree. On the other stages, it can be as low as in 12% of agreement between the tracers.
All right, let that sink in. These 2 FDA-approved scans, both designed to detect the same tau pathology using the Brack staging system, which is the gold standard for tracking how tau spreads through the brain, well, they agree less than half the time. In some brain regions, particularly in early Bragg stages, where intervention might be the most effective, they only agree 12% of the time. That's not a minor discrepancy. That's a fundamental disagreement about what's happening in the brain. But here's where it gets even more interesting. Dr. Roca has discovered why they disagree.
When we isolate these discordant individuals, we identify that in 87% of the cases, the reason for the disagreement was because MK was assigning a higher Bragg stage. This signal was genuine tau spread signal. So what seems to be happening is that MK is always one step ahead.
So MK6240 isn't just different. It's detecting tau pathology earlier, potentially catching the disease when tau is still in its nascent, less mature form. And this isn't a technical error or an artifact. 87% of the time when they disagree, it's because MK6240 is detecting tau that Flortaucipir simply can't see yet. During the Q&A session, Dr. Roca explained this critical difference.
If you're gonna actually use a threshold to try to differentiate it from the background, you don't differentiate. In MK, you can differentiate it very easily, and you have a lot of patients that don't have any signal FTP and already have a very clear abnormal signal in MK.
So these implications are enormous for early interventions. If we're using Flortaucipir, we might miss early tau accumulation that MK6240 would catch. So tau that's already beginning to damage our neural network but hasn't yet formed into the mature tangles that Flortaucipir detects. Dr. Roca revealed just how much earlier MK6240 detects tau.
She did the same thing I did, but she used centiloid like a timeline. So you can see in which centiloid MK becomes positive and which centiloid FTP becomes positive. There's a gap of 20 centiloids or more, 30?
So 20 to 30 centiloids in the language of amyloid PET imaging, that represents about 3 to 5 years of earlier detection. Years where intervention might be more effective before tau has spread extensively through our neural network. Quick break here. If you are finding value in understanding these breakthroughs in imaging discoveries that could basically change how we detect Alzheimer's years earlier, well, make sure to subscribe to this channel. I translate the latest research conferences into actionable insights for us APOE4 carriers and anyone concerned about their brain health. Hit that subscribe button so you don't miss out on our next deep dive into game-changing research. Cool, let's shift to another groundbreaking discovery. Dr. Ismail Kalandri's work, presented by Dr. Sophie Steinberg from the Lund University, proves something clinicians have long suspected. Alzheimer's doesn't follow just one pattern of brain atrophy. Using a sophisticated machine learning algorithm called SUSTAIN on over 2,000 brain scans, they identified distinct patterns that persist from diagnosis through autopsy.
We identified 3 distinct MRI subtypes. The first showed a limbic predominant pattern with early atrophy in limbic regions. The second subtype showed a more posterior pattern. And the final subtype had early cortical atrophy, but only in very late stages the hippocampus got affected. So this is hippocampal sparing subtype.
So 3 completely different patterns of brain shrinkage, each telling a different story about how Alzheimer's unfolds. The limbic predominant pattern attacks our emotional and memory center first. These patients often show classic memory loss. The posterior pattern affects visual and spatial processing areas, so these individuals might struggle with navigation or recognizing objects before memory problems emerge. And then the hippocampal sparing subtypes preserve memory centers until very, very late, but attacks executive function early. So these patients might maintain memories but lose the ability to plan or make decisions. But here's what makes this finding truly revolutionary. These aren't just imaging patterns. They correspond to actual pathology found at autopsy.
The posterior subtype had more p-tau in parietal regions than the others, whereas the posterior and limbic subtypes had more p-tau in middle frontal and temporal regions. The limbic subtype had more NFL in temporal and parietal regions.
So p-tau that we covered in the previous video, phosphorylated tau, is the toxic form of tau protein that forms those tangles. NFL, which are the neurofilament light chain, is a marker of these neuronal damage that can now be measured in blood tests for around $300 to $400 at major labs. This different distribution validates what many families have observed, that Alzheimer's can look different in different people. Some lose the memory first, others lose visual processing or the language. Now we know these differences reflect genuine biological variations in how the disease progresses, potentially requiring different therapeutic approaches for each subtype. Now we come to perhaps the most counterintuitive finding of the entire conference. Dr. Ting Tri from McGill University, analyzing data from the PREVENT AD cohort, which is a study that followed people at risk for Alzheimer's for over a decade, discovered something that contradicts everything we thought we knew about brain changes in Alzheimer's.
Cortical thickness may increase in the early stages and decrease as the disease progresses.
Wait, brain thickness increases? This flies in the face of decades of research showing brain shrinkage in Alzheimer's. But Dr. Tri's team analyzed over 10 years of longitudinal data, tracking the same individuals as they progress from healthy to at risk to diseased, and found something truly remarkable.
In the amyloid-negative individuals, higher amyloid was linked to greater volume, greater thickness, and decreased mean diffusivity, especially in the parietal and the medial temporal regions. In the amyloid-positive group, more amyloid was linked to lower volume, lower thickness, and increased mean diffusivity.
So rugosity refers to the brain surface complexity. It's basically the folds and the wrinkles that you can imagine your brain has. Before crossing the threshold for amyloid positivity, which is approximately 20 to 24 centiloids on amyloid PET scan, the point where someone is considered to have Alzheimer's pathology, while increasing amyloid is actually associated with larger brain volumes and smoother surfaces. Only after crossing that critical threshold does the expected shrinkage and increased folding begin. So this kind of paradigm shifting research is exactly what we discuss in depth at the Phoenix Community, where we help APOE4 carriers like you and me understand not just what's happening in our brain, but when and why it matters for prevention strategy. So if you're not a member yet, the link to apply is in the description below. Next, Dr. Chu pinpointed exactly why this reversal and when this reversal happens using sophisticated timeline modeling.
We aligned MR data based on their cell estimate years from amyloid positivity, and we found brain volume and cortical thickness gradually increased in the years leading up to the amyloid positivity, then declined afterward, showing a clear biphasic pattern.
So the biphasic pattern, growth followed by shrinkage, basically, suggests our brain undergo a critical transition period that we've been completely missing with traditional imaging approaches. But what causes this paradoxical brain enlargement? Well, Dr. Chu offers 3 possible biological mechanisms.
This earlier increase in volume or thickness could reflect inflammation, cell swelling, or the physical space taken by the plaques before neurodegeneration starts.
Think about that. Our brains might actually be mounting an inflammatory response with immune cells like the microglia rushing in and causing tissue swelling as they try to clear the toxic proteins. Or neurons themselves might be swelling as they struggle to maintain function despite accumulating damage. Lastly, and another option is that the plaques themselves, those clumps of amyloid protein in your brain, might be taking up physical space between neurons before the cells start dying. This completely changes our understanding of the disease timeline.
Our findings suggest structural changes start years before amyloid positivity, and they follow age-dependent trajectories.
So for us APOE4 carriers, this biphasic pattern might be even more pronounced or accelerated given our enhanced inflammatory responses and altered amyloid clearance. Understanding this window when our brains are fighting back but haven't yet succumbed could be crucial for timing the intervention. So understanding these complex brain changes is one thing, but analyzing them at scale is another challenge entirely. So Dr. Amir Desarvi from LMU Munich presented AdPrep, a fully automated pipeline that's removing the technical barriers to advanced neuroimaging research. This containerized software, meaning it runs consistently across different computer systems, can process multiple types of brain scans, including structural MRI, functional MRI, PET scans, and diffusion imaging.
AdPrep has already demonstrated significant impact in real-world research, contributing to over 25 research papers. We have successfully processed neuroimaging data from thousands of participants across large-scale data sets, including ADNI A4 ppmi HABs with an impressive overall processing failure rate of less than 4%.
So a 96% success rate across major Alzheimer's cohort, including the ADNI cohort and the A4 cohort. Processing times like these are remarkably fast, about 1 hour for combined structural and functional MRI and just 30 minutes for PET scans. And the accuracy is truly remarkable.
We have rigorously benchmarked ADPrep using data from the PET core. Our pipeline closely reproduces amyloid PET SUVRs with a correlation of 0.99 and tau PVRs with a correlation of 0.98.
So near perfect correlation with the gold standard measurements. So centiloids are the standardized scale for measuring amyloid burden, while SUVR measures how much radio tracer accumulates in different brain regions. This means smaller research centers without specialized imaging expertise can now perform the same sophisticated analysis as major institutions, which will potentially accelerate discovery for all of us by years. Finally, the last topic is addressing the practical problem. So Dr. Emma Ruppert from the University of Pittsburgh basically solved. Given that different tau tracers show different patterns and pharmaceutical companies are increasingly using MK6240, how do we ensure consistent interpretation across different scanners and readers?
Disagreement for Flortaucipir, 26%. The harmonized method had a disagreement of 11%. Similarly, for MK, disagreement was cut from 23 down to 8%.
Well, her team developed a harmonized visual reading method that dramatically improves agreement between different radiologists reading the same scan, cutting the disagreements rates by more than half. This means when we get a tau PET scan, we can have much more confidence that the interpretation is accurate. But more importantly, it helps reconcile differences when comparing the two tracers.
The disagreement for Rater 1 went from 32 down to 12%, and for Rater 2 went from 26 down to 16%.
This harmonization is crucial because tau PET scans aren't just the research tool anymore. They are becoming part of clinical practice and drug trials. In fact, I am sure a lot of you watching these videos have done their own tau PET scans. So during the Q&A of the session, Dr. Ruppert provided crucial insights into why Flortaucipir can be so challenging to interpret.
It's something that does show up, and it doesn't show up in MK. MK, it's easier to be sure that it's positive or negative, because when there is uptake, it's very clear. And with Flortaucipir, we see this kind of yellow temporal binding that is inspecific. It is something that once you play around with the range, usually it disappears, which doesn't usually happen when there is high uptake.
So this non-specific binding in Flortaucipir, which is essentially background noise that can be mistaken for real tau, well, this background noise doesn't occur in MK6240, making it a cleaner, more reliable signal. For those of us tracking our brain health over time, this clarity could mean the difference between catching problems early and actually missing the window for interventions, which sometimes can be quite tight. So this conference session concluded with a powerful observation from industry consultant Jeff Hawk that tied everything together and has immediate implication for clinical trials and future treatments.
I would point out that if you look at the trials the pharmaceutical companies they're using one tracer, and it's the one that's ahead, MK. Some of the information that you get from MK, you don't get from FTP, and you can't get it back. How do you get that information, that early information, those differences that you're seeing, how do you get it back?
Pharma companies developing the next generation of Alzheimer's drug have made their choices. They are using MK6240 because it captures information that Flortaucipir misses. And once that window passes, basically those critical years when tau is accumulating but hasn't yet formed mature tangle, well, the data is gone forever. So this has profound implications for clinical trial enrollment and treatment monitoring. Dr. Roca offered a final thought on why these tracers might show such different results, suggesting they might be binding to fundamentally different forms of the tau protein.
I wish we had cryo-M of all of them to know exactly which part of the tangle they are binding, different of the tangles, because sometimes it looks like one is binding to a more mature tangle, the other one is binding to a less mature tangle, because you can see that the signal is overlapping but not that much.
For cryo-electron microscopy would reveal at the molecular level exactly which part of the tau protein each tracer attaches to. They may actually be detecting different stages of tau tangle formation. For example, MK6240 are catching earlier, less mature tau tangles that are still forming, while Flortaucipir only detects more developed pathology when the tangles have fully formed. So the difference isn't just academic. It could really determine whether to catch the disease when it's still reversible. So these imaging breakthroughs fundamentally changes how we think about Alzheimer's detection and progression. What we've learned today is that tau tracers disagree more than they agree. With MK6240 detecting pathology potentially 3 to 5 years earlier, we've discovered that brain paradoxically enlarge before they shrink, and potentially from inflammation or the physical bulk of accumulating proteins, which is a critical window we have been missing. We now know that there are 3 distinct patterns of brain atrophy, each with its own pathological signature and clinical presentation. And now we understand that our brain's waste clearance system, so those perivascular spaces that clean our brains while we sleep, may be the key to understanding cognitive decline. So for those of us who are APOE4 carriers or in the Phoenix community, navigating this complex landscape, these findings reinforce a crucial message. The window for detection and intervention is wider than we thought, but we need to use the right tools at the right time. The revolution in brain imaging isn't coming, it's already here, and it's changing everything we thought we knew about preventing Alzheimer's. So stay informed, stay proactive, and remember, knowledge is always our first line of defense.